So, hello everybody. I'm very glad to be here, and I would like to thank the organizer to give me the opportunity to share my data with you. So, I'm a biologist, and today the goal of this presentation is, there is two actually. One is to convince you that you have to study the non-tumor cells surrounding the tumor cells, and these surrounding cells are perhaps as important as the cancer cells. And the second one is to promote discussion about my model, and I will be really happy that in the end of this talk you come to see me and say, okay, I would like to include your model in my mathematics model. So as Christine said, you can find me in the Institute for Research on Cancer and Aging in Nice. That is a city that is very close to here. So in our team, we are working on from the initiation of the cancer through the metastasis and the detection of circulating tumor cells in the blood. And for this, we are many people in the team. So there is basic researcher, but also physicists, so pathologists and oncologists. And so basically we can start from a molecular model to the discovery of new biomarker in patients due to the biobank aided by Paul Hoffman. So today I will talk like some other talks about lung cancer. It's a leading cause of cancer deaths in France and worldwide, poor survival because of the late diagnosis. Most of the patients will have already metastasized. But also there is like few treatment. Now there is immunotherapy, as you listened to this before. But today we'll focus on stage 1 patients. So stage 1 patient is patients that have a very small cancer, and that they will go for surgery, but after a while, like 5 years, 10 years, most of them will relapse. So it's a huge problem to know which patient to follow and which patient to add some treatment after the surgery. Why I will try to convince you that non-tumor cells are really important in the tumor. It's because of this graph. If you look at this, this is different tumor, and this is the purity of tumor cells within a tumor. So meaning that this is the percent of tumor cells within a tumor. And you can see that it's really heterogeneous. So, and lung cancer is perhaps the worst one. It's meaning that there is, in some patients, only 20% of tumor cells within the tumor, cancer cells within the tumor. And for just here, it's for example melanoma. Here is breast cancer. Okay? So basically, more a cancer is diluted within surrounding cells that are not cancer tumor cells, not cancer cells, but then it's a really aggressive disease. Okay? So what are inside the tumor? There is many things. There is cancer cells, but there is also normal cells that will compete for nutrient, oxygen, and in confrontation with the tumor cells. Also, there is fibroblasts, and there is vessels, and there is also all the immune system, all together mixed in one organoid, we'll say. Today, I will focus my talk about two parts. One part is a small story that we published a few years ago about the role of neutrophils and the modification that the neutrophils could promote in the cancer cells that will undergo into a EMT program. The second one is a project that will try to have an overview of the entire tumor. Okay? So the first one, this is something that we found in vivo. So basically, it's a pathologist that is in the team, and what he discovered is that more neutrophils there is in a tumor, the higher chance you have to relapse. Okay, so this is a normal trend, and he put a mark, and he said, okay, what is below and higher? So if you have a higher neutrophils rate, then you have a higher chance to relapse, and if you have very low neutrophils, you won't relapse. And we did also blood analysis, and we found out that a microRNA, I will explain you later what is a microRNA. So just take it like a marker in the blood, and we saw that this microRNA was associated with a relapse of the patient. So what are microRNA? MicroRNA are very non-coding RNA, very small one, and that will bind to a protein that is called AGO, and all together they will bind to mRNA. The basic system of this is that this complex will block the translation of the mRNA. Basically, if the microRNA bind to the AGO and bind to the mRNA, there is no translation, there is no protein anymore. So we could quantify the microRNA, and microRNA 223, is mainly expressed in neutrophils and not in other cells in the body. So basically, as I told you, there is neutrophils that are important, but in the blood we can detect an increase of the microRNA 223. And some microRNA are very specific, as I told you here. So basically, if we look at these two graphs, you can say that perhaps microRNA 223 is pro-tumoral and associated with poor survival. How to quantify the efficiency of a microRNA? A microRNA, as I told you, will block the translation of mRNA to the protein. So basically, we can follow the activity of a microRNA by looking at the target. And here we choose this one because it's clear-cut. We have nice differences between the two. But also, as a microRNA may have many targets, we could follow also a phenotype. And here the phenotype is that we use a chamber where you look at the migration of the cells. So basically, in this chamber, there is a matrix, a fibronectin matrix, and a filter, and the cells has to pass through this matrix, through the filter, and we only count the cells that are past all this change. So basically, here we saw that the microRNA 223 increase the rate of migration of the cell. So in the few talk, we will talk about EMT. So what is EMT? Is like we can induce EMT by TGF-beta, so there is an increase of some transcription factor here, and you see that microRNA 223 is able also to do the same, but also there is a vast change in the cells. So the cells, basically, ikaderin is a marker of EMT, the marker is expressed at the membrane. Upon TGF or microRNA, there is no expression at the membrane anymore. And there will be a remodeling of the actin cytoskeleton. So here is stress fiber, and the microRNA 223 induce philopodia. And 2 days ago, if you remember, there was a person speaking about the role of philopodia in the migration of the cell. But, and there is a but, if you incubate for many days microRNA 223 in cells, there will be a cell cycle arrest and cell death. So in some paper, in vitro, you will find that microRNA 223 is an anti-tumoral microRNA. So wait, it's exactly the reverse of what I said before, that we have some proof that microRNA is pro-tumoral. So here we have a problem.
But the micro-23 in neutrophils stays in neutrophils.
No.
Does it go out?
I will show you later. Okay. So here we have, so we start to think that we have a problem of dynamics of the microRNA, and so perhaps the microRNA is not always expressed. So sorry, this is a neutrophils, and this is a cancer cell, sorry for the transfer, and we start to think that perhaps the microRNA could be transferred from the neutrophils to the cancer cells. Sorry. So basically, here are neutrophils, here are cancer cells, in vitro cancer cells, and this is the level of microRNA that we could detect. And actually, it is in log 2, so basically, in neutrophils, there is 30,000 times more neutrophils than in cancer cells. Basically, there is no expression of microRNA 223 in cancer cells. What we show is that if we co-culture them, then there will be, you could detect the microRNA 223 within the cancer cells. So meaning that there is a transfer, to answer your question. There is a transfer from the neutrophils to the cancer cells of microRNA 223. And this is not a burst of transcription, because we block the transcription and there is still some transfer. So it's not that the cancer cells start to express microRNA-223. No, it's a real transfer from the neutrophils to the cancer cells.
So this is with the exosomes? Or?
Yeah, exactly. So we discovered that it's supernatant and it's extracellular vesicles. But what we do also is try to see what was the dynamics of that. And the dynamics, so overnight we incubate the neutrophils with the cancer cells. So we load the cancer cells with microRNA-223, and then we remove the neutrophils. And here is perhaps one of the best results I have in my career. Okay, so you see a decrease of microRNA-223. And this is very nice because most of the time you are using synthetic microRNA, and that are not you cannot destroy into the cells. So here it's natural microRNA, and you see a decrease of the microRNA. And if you look at the target, FOXO1, here is exactly the opposite. The protein is restored over time. So meaning that since the microRNA is decreasing, the protein is recovering. And what about the phenotype? If you took these cells, they could migrate. And if you took these cells 8 hours after the washes, so no microRNA anymore, they could not migrate anymore. So meaning that there is a very rapid switch between the two states from a migratory cells and a cell that is not able to migrate anymore within 8 hours. So we wanted to know a little bit deeper about the mechanism. And actually, there is some enzymes that are known to degrade mRNA. So basically, there is two big families, the XRN1 family that will degrade mRNA from 5' to 3', and the D3 family that will degrade from 3' to 5'. So we did a screening assay, and we found out that the XRN1 enzyme was able to maintain a high level of microRNA-223 within the cells, and even you can see here at the loading step there is a little bit higher level of microRNA within the cell. Well, so what about the kinetics? So then if we invalidate the XRN1, you can see that we are not able to block completely the degradation of microRNA, but we delay the degradation. And the direct associated phenotype is that the cells are able to migrate a little bit more. So I don't know if there is any consequences. So it's difficult to say if there is the correlation. But what we found also, it's in 25% of the patient of lung cancer, there is a deletion of the gene of XRN1. And the patient who have the deletion of the genes, they will have a tendency to relapse more. So I don't know if it's associated with the fact that they are not able to degrade microRNA-223. Perhaps there will be also other mechanisms, but it's nice to have this kind of correlation. So basically for this first part, we discovered that there was a rapid decay of the mRNA and microRNA that is going from outside into the cancer cells. There is a very rapid switch in phenotype. So actually, it's very quick, and for EMT person, I think that it will be funny to discover that within 8 hours they could switch from one state to the other one. There is an involvement of XRN1 in the decay. Of course, the diffusion is really important because if the neutrophil is not close from the close enough to the tumor cell, then there will be less microRNA that will arrive to the tumor cells, so perhaps they will migrate a little bit less. And of course, they give you the idea about the role of neutrophils in cancer progression and the role in metastasis. Okay? So, given the fact that I've seen some mathematics here upon this last 2 days, I think that there is some parameter that could be easily translated in mathematical processes, and I would be really happy to discuss with you about the way to do it. So now the second part is about to have to work about the overview of the tumor. And the idea behind this is, as Christine mentioned, is that now we have access to molecular data, and it's easy now to do a transcriptomic analysis. So the basic idea is it possible that we have a bulk of mRNA, and by bioinformatics, by mathematics modeling, we are able to separate from this bulk different signatures of different cells. And that is not that easy, but it's something that I would like to do because I have the unique signature of each of them. Okay? So for math aspect, it will be to define the best model. For biological aspect, it will be to reduce the variable. Like Victor said a few days ago, if you change of dimension, it doesn't mean anything for me. If you change of dimension, it will be helpful for you, but for me, it doesn't mean anything. So I cannot verify if the way you find it, it will be easier. So that way, I would like to stick to the percent, I will say at the end, of the different families, so I can say that, okay, perhaps in the biopsy, I could find exactly the same number that you will give me, okay? And after, it will be helpful for the pathologist, because then he will confront the vision he has on the slide and the vision of the transcriptomic data to stratify the patient and perhaps to follow them better. So as I say, what we did, we started with a patient, and we have cancer patient, and we have all the different components of the tumor, and we tried to stick on the same microarray, so the same platform for the analysis, because then after it's too complicated. So what is a microarray? So this is because perhaps you have never heard about this, so I want to be as clear as possible. So this is a microarray. On this little square, there is billions of genes that are on little cell, I would say, and each cell is for one gene, or at least there is many cells for one gene. So here you quantify the level of signal, and it's going from 0 to 65,000 level of gray. And more the cell is bright, more there is a signal, and more the gene is expressed. Okay? So as you can see, there is a lot of blue, meaning that there is few signal. So here is exactly what you see. Here I spot the intensity of signal in log 2, and here is the number of genes that are in each category. So basically you see that this is noise, and this is genes that start to be expressed. So if you give that to persons that do machine learning straight, they will work basically. So what I say, I try to remove the noise and the housekeeping gene. Housekeeping gene is the gene that has no variation between all the patients. So basically we end up with 4% of the gene. But if you give to people that are doing machine learning, deep learning, whatever, all this list, they will find, sure, a nice separation between the variables. But most of the time they will work either on noise or on housekeeping gene. So there was no signification at all for the patient. So here we want to focus on this 4% of gene of interest. So this is exactly the same thing. So this is pure population. This is for patient. You see that it's not exactly the same, but at the end we have a nice curve so we can interpret the data. But here, it's my problem. Right now, it's a mathematical problem that I have. Is that if you look at subpopulation, the subpopulation are almost twins. It's really tricky to differentiate a CD4 lymphocyte from a CD8 because they have 98% correlation. So if you want to do partial least square or least square, it's very tricky because then, as you know, for mathematicians, they have to be independent variable. And these are not at all independent variable. So this is tricky. We have to reduce the number of genes, perhaps, to reduce the number of correlation. Or what we decided also is to merge some of them that we know and make one unique population from the one that we could not differentiate. But also what I did is I did it manually. So basically, as I have the pure population, I say I know the genes. Okay, this is really a gene that is expressed in neutrophils, for example. So I know the level that is in the pure population. So I will guess, if I look at in the patient, what is the level of these neutrophils in the tumor. And I have to say that when we confront these two data, it's completely different, okay? Anyway, so we managed to have like some simulation. So this is the calculated intensity by a math method and what we observe in terms of intensity for the patient. So there is some correlation, but some of the other ones are not good. So, and this is, I have to say, the best patient. So, okay.
What does this mean again?
So this is what we calculated by least square or partial least square. Okay, that is the coefficient we use. We add a coefficient for each population, and we reconstructed the entire bulk. And then we look at what was the calculated intensity that we got by mathematician modeling and the observation we have in the tumor. So it's pretty cool, but I have to say it's the best patient. But eventually we got this signal. And this is fibroblast, epithelial, vessels, CD4, so monocyte, B cells, CD8, neutrophils. And I give that to pathologists and say, okay, is it something that what's wrong with this? And he say, okay, fibroblast, there is a huge fibroblast reaction in the tumor, so that could be fine. Here, as epithelial is perhaps a little bit low, because as you show in the purity, there is from 100% to 20%, so perhaps this is a little bit low. And here we have a huge B cells signature that we cannot find, or not so much B cells in the tumor of patients. So this is something that we don't understand yet so far. So, but we have to manage this. I don't know how to do, but we will try to find a way. So basically, I focus my attention about the CD8 and the neutrophils. The neutrophils because it's my population of interest, and CD8 because it could be interesting for immunotherapy. So we cross our math model with the clinical data we got for this patient. And actually it's kind of cool because we have some results that fit with reality. So actually more the staging is increasing, less you have lymphocytes for two different types of cancer, of lung cancer, and more the cells, because the tumor cells increase in staging, more there is neutrophils. So it's a good tendency, and I will say that it's pretty cool. So now what we did, but because some people do that in publication, we make a ratio between neutrophils and lymphocytes, and we look at the survival of the patient. And actually, like the first thing, we show that more the ratio is high, more the patient have a tendency to relapse earlier and to die earlier than the other one. And so it's exactly the first paper that I showed you that the neutrophils are important for things. So I think that there is a correlation, but we have like a black box still that we have to improve the math modeling between the bulk information and the patient of data, because we know that as a proof of concept, it could work. But we have to improve this. So I would like to thank you for your attention. I would like to thank all the people that helped me for the study, and special thanks for the Canceropol that funded me for the two projects. Thank you very much.